Good morning and welcome to Parasitology and Mycology. My name is Dustin Scott Brewster, and today is part 3 of the malaria and other blood and tissue protozoa. The first of 4 species of Plasmodium that we're going to talk about is benign tertian malaria. Benign because it is less severe than other species of Plasmodium, and tertian because of the paroxysm that occurs every 3 days as a result of the length of its asexual cycle. The stages present on the peripheral blood film can all be present, and not one stage is dominant over another. The size of the infected red blood cell is typically enlarged with Plasmodium vivax, and in the developing young trophozoite, as we see here in the upper image, you can see the irregularity in the shape of the trophozoite. Stippling is usually present at a pH of 7 to 7.2 using a Giemsa stain, and in the more developed trophozoite, like we can see in the bottom image here, the density of the cytoplasm is diffuse and therefore results in light staining. Pigment can be present in older organisms that appear blackish brown to yellowish black, and that we can see in the upper image here in an older developed trophozoite with pigment present. And the average number of merozoites is around 16. This is a mature schizon. It looks like it's ruptured already, so it may not be an accurate count of the number of mature merozoites in the schizont, but Plasmodium vivax averages around 16 merozoites, which is a species-specific number. The length of the asexual cycle is 48 hours, resulting in a paroxysm every 3 days. Prepatent period is 8 to 17 days, and the incubation period is 14 days. Next we have Plasmodium ovale, known also as benign tertian malaria. Again, benign because it's less pathogenic, and tertian because of the paroxysm that occurs every 3 days. To distinguish the common name benign tertian malaria from Plasmodium vivax, this species is also known as ovale tertian. All stages can be present in the peripheral blood smear, but usually one stage is dominant over another. Just like Plasmodium vivax, the size of the infected red blood cell for Plasmodium ovale is also enlarged, but different because the infected red blood cell typically has this oval shape and fimbriated edges that you can see here and here. So oval shapes. Stippling can also be present at a pH of 7 to 7.2 using a Giemsa stain. The density of the cytoplasm is compact, resulting in a darker staining characteristic, and the average number of merozoites in the mature schizont is around 9. The length of the asexual cycle is 48 hours, again resulting in a paroxysm every 3 days. Prepatent period is 8 to 17 days, and the incubation period is 14 days. Next, we have Plasmodium malariae, known as quartan malaria. All stages can be present, but one is typically dominant over others. The size of the infected red blood cell is normal or slightly smaller, and it has this characteristic band shape. The trophozoite has this characteristic band shape, which would represent Plasmodium malariae. Stippling is not present with Plasmodium malariae. The cytoplasm is compact and results in a darker staining. Pigment is usually present in older organisms, appearing dark black. The average number of merozoites in the mature schizont is around 9. The length of the asexual cycle is 72 hours. This is unlike the last 2 species we talked about, so you would have a paroxysm every 4 days instead of every 3 days. Prepatent period is 14 to 30 days, and the incubation period is 30 days, so a longer incubation period.
Let me think about that. Okay.
Next, we have Plasmodium falciparum, known as malignant tertian malaria. Malignant because it is more pathogenic, more severe than the other 3 species of Plasmodium we just talked about. And tertian, again, because of the paroxysm that happens every 3 days. All stages can be present, but it's typically just the ring stage or the gametocyte stage of the organism. So rarely you'll see other stages of Plasmodium falciparum. The size of the infected red blood cell is normal, so you won't see an enlargement like you will on the first two species we talked about. You can see stippling. It's rarely present, and a unique feature of Plasmodium falciparum is a staining characteristic known as Mauer's clefts. The density of the cytoplasm is compact, which would result in a darker staining characteristic. Pigment can be present. It usually appears black. The average number of merozoites in the mature schizoid is around 24, so far more merozoites than the last three species of Plasmodium that we talked about. This would result in more red blood cells being infected, more red blood cells being lysed, and a more severe case of anemia and malice that would result in symptoms such as black water fever that we talked about in one of the first lectures. The shape of the gametocyte is unique in Plasmodium falciparum. It is a banana or sausage shaped. We can see in this bottom image here that is a very telltale sign of Plasmodium falciparum. The length of the asexual cycle is 48 hours. The prepatent period is 5 to 15 days, and the incubation period is 12 days. Here we have a chart to help distinguish between the four species of Plasmodium that cause human infection. On the left, we have the presence of enlarged infected red blood cells. On the right, we have normal infected red blood cells. So if we have an enlarged infected red blood cell, it would lead you to believe it's Plasmodium vivax or Plasmodium falciparum. The next thing I would look at, I would go down to the average number of merozoites in the mature schizoid and average those out, and that would stick you to one of these columns here. I look at a couple of other signs. Is the mature trophozoite, does it have a compact or diffuse cytoplasm? How lightly does it stain? Is the developing trophozoite irregularly shaped, or is it oval? And you can deduce which species of Plasmodium is present based on those characteristics. Next, we have the other section of the blood and tissue protozoa, and up first we have Trypanosoma cruzi. And infection of Trypanosoma cruzi begins with not the bite of the reduviid bug, but when a reduviid bug bites a human host, it releases at the same time metacyclic trypanosomes in the feces near the site of the bite. The human host would then scratch in the trypanosome infected feces into the site of the bite, or infection can also occur through direct contact of the metacyclic trypanosomes into the mucosal membranes such as the eye. Once the metacyclic trypanosomes enter the human host, they will infect cells near the site of infection and develop into cellular amastogotes. These cellular amastogotes will divide by binary fission and break free of the cell near the site of infection and enter the circulating bloodstream as trypanosomes. These trypanosomes will then travel and infect new tissue, again repeating the cycle and developing into more amastogotes, going through binary fission and perpetuating the cycle in the human host that we can see here. Also, the reduviid bug can come along and take a blood meal from an infected human where it was not previously infected and ingest the trypanosote phase of the parasite. Inside the reduviid bug, the trypanosote will travel to the midgut and develop into an epimastogote where it will reproduce by binary fission, and the epimastogote will then produce more epimastogotes, which will develop into metacyclic trypanosomes in the hindgut, where it will be passed in the feces as it takes a blood meal from its next victim. Again, transmission from T. cruzi in humans is passed on by the reduviid bug, not from the bite itself, but from the feces of the reduviid bug containing the metacyclic trypamastigotes, and that metacyclic trypamastigote feces being scratched into the bite, or through direct contact from the mucosal membrane entering the host, such as the eye. The results of the T. cruzi infection in humans produces American trypanosomiasis, or Chagas disease. Again, the bite of the reduviid bug does not transmit the disease. It's from the contaminated feces being scratched into the bite site, resulting in American trypanosomiasis, also known as Chagas disease. And the morphology present in the upper image here, we have circulating trypamastigote, and it has this characteristic C shape with this nucleus in the middle third. This nucleus in the middle third of the body, and a terminal kinetoplast that serves as an attachment for the undulating membrane that lines the organism. And off the anterior end, we have the flagellum that it uses for locomotion to infect new cells and develop into the amastigote phase. That would lead us to the bottom image here. We have a biopsy of infected cells and amastigote tissue phase, which measure 3 to 5 microns in diameter and contain a nucleus and a rod-shaped kinetoplast. In humans, we have the trypamastigote and the amastigote form. The amastigote form, again, is the tissue phase. The trypamastigote stage is the blood stage. Epimastigotes are the developmental stage, found only in the reduviid bug reservoir. The acute phase of Trypanosoma cruzi can last 1 to 4 months in duration, and the chronic phase can last years to even decades. Clinical signs of T. cruzi can result in fever, headache, malaise, megacolon, and megaesophagus, as well as cardiac manifestations from the amastigote tissue phase. In the event that infection does occur through crossing of the mucosal membrane, you get severe edema of the eyes from the cells near or around the site of the eye being infected with the amastigote phase of the parasite, and this is known as Romania sign. Toxoplasma or Trypanosoma cruzi is most prevalent in South America, especially Brazil, Argentina, Uruguay, Chile, and Venezuela. Cases have been documented in Central America in states such as Texas and California, resulting in a concern for blood banking. Many animals can serve as reservoirs for Trypanosoma cruzi, resulting in dogs, possums, cats, and armadillos carrying the parasite. Next, we have Toxoplasma gondii that I've said several times now by mistake. And the only definitive hosts for Toxoplasma gondii are members of the family Felidae, which are domestic cats and their relatives. These infected cats shed large numbers of unsporulated oocysts for 1 to 2 weeks in large numbers. These unsporulated oocysts become infective and sporulate in 1 to 5 days in the environment, where they can be found in food products, water, or litter boxes from infected cats. Humans get infected through ingestion of these sporulated oocysts, or through ingestion of animals that have been infected with the sporulated oocysts. Once the animal ingests sporulated oocysts, they develop into tachyzoites, where they navigate themselves to neural and muscle tissue, where they develop into bradyzoites, and they stay there in the tissue of the host. And humans can come along and ingest uncooked or undercooked meat products, ingesting these bradyzoites and becoming contaminated and becoming infected through this route. So ingestion of bradyzoite cysts from animals ingesting the sporulated oocysts, or humans can become infected directly from contaminated soil, food, and water, or litter boxes from sporulated oocysts directly. The definitive host is the domestic cat, which is of most importance. Ingestion of infected oocysts from these the cat's feces can contaminate litter boxes, and most humans get infected that way. This would be ingestion of the trophozoites and undercooked. This can also occur through the ingestion of the trophozoites in undercooked or uncooked meat, or from ingestion of breast milk passing on from a mother to a child. Symptoms of Toxoplasma gondii are usually asymptomatic. The immune system does a good job of keeping this infection in check, and clinical symptoms usually only manifest themselves in patients that are immunocompromised, such as people going through chemotherapy, or individuals with immune diseases such as HIV. It is particularly dangerous in pregnant women because of the organism's ability to cross the placental barrier and infect the developing fetus. Diagnosis of Toxoplasma gondii is done through tissue biopsies of the cysts, the bradyzoites, through serological techniques, or through DNA analysis of the amniotic fluid. Last, we have Pneumocystis carinii, which isn't technically a parasite. It is classified as a fungus, but the staining techniques are done in the parasitology lab. The infection of Pneumocystis carinii results in interstitial plasma cell pneumonia, which is a very fatal complication of AIDS. The organism is widespread, and similar to toxoplasmosis, the body does a good job of regulating the infection, and those that have a compromised immune system are at greatest risk for developing the interstitial plasma cell pneumonia. Diagnosis of Pneumocystis carinii is through identification of the cysts containing the 8 trophozoites, or from the free trophozoite forms in lung exudates, sputums, open lung biopsies, or brachial alveolar lavage samples. In the top image here, we have a Giemsa stain that shows the spherical bodies and the 8 internal trophozoites, but it won't show you the cyst wall. In the bottom image, we have a methylene silver stain that allows the cyst wall to be seen, but not the internal structures. So both of these stains are used to complement the identification of Pneumocystis carinii. That concludes the third part of malaria and other blood and tissue protozoa.